Hello everyone. So today we will learn about the Eimeria parasite, which come under the phylum Epicomplexa, class Sporozoa. Member of the Sporozoa class are parasitic in spore forming, and phylum Epicomplexa has a apical complex, that is polar ring, conoid, micronemes, rhabdoids, micropores, and subpellicular tubules. So both asexual, that is schizogony, and sexual, that is gametogony types of reproduction are present, and member produce spores by means of sporogony. Okay, this one is important. Now it comes under the subclass, that is coccidia, order Eucoccidia, family Eimeridae, Cryptosporidae, and Sarcocystidae. So organism commonly called as coccidia. It is intracellular parasite found in the epithelial cells of intestine of vertebrate, in which both asexual and sexual reproduction take place. So it is present in the epithelial cells of intestine. It is highly host specific as well as the organ or location specific parasite. Okay, and the cycle is direct life cycle, that is it doesn't need any intermediate host, and locomotion is by gliding. Okay, now unsporulated oocysts come out of the infected host through feces, and sporulation of the unsporulated oocyst occur outside the host and environment. Okay, so this is important, that sporulation will be formed outside the host in the environment. And sporulated oocyst is the infective stage for the host, and this one is also important. This sporulated oocyst is the infected stage for host. Okay, so we will here talk about the unsporulated oocyst. It is spherical, subspherical, ellipsoidal, or oval shaped. Okay, usually bilayered, transparent, and clearly visible. Small pore called micropyle may be present at narrower end, which is often covered by a polar cap or micropylar cap. A nucleated mass, that is sporont, is found mostly at the center. So here in the picture, you can see the narrow end. All right, this pore is called micropyle, and a cap is present here called polar cap. Okay, this is dense nucleated mass. It's called nucleated mass called sporont at the center. Okay, this is the sporont. This is the structure of unsporulated cyst, and this unsporulated cyst come out of the infected host through feces. Okay, so if we take the sample of feces, you will see this unsporulated oocyst. Okay, sporulation of the unsporulated oocyst occur outside the host and environment. Okay, so sporulation occurs in the environment. Okay, now this is structure of sporulated oocyst. So sporogony occur outside the host, and sporulated oocysts are the infective stage of the parasites. Okay, time taken for sporulation can also help as an aid to identification of species. Okay, and sporulated oocysts contain different numbers of banana-shaped sporozoids. Okay, these are the sporozoid. You can see here the banana shape, right? Here we can see this banana shaped is sporozoid with the nucleus, sporocyst residuum, and stida body. Okay, sporocyst residuum and stida body. This is the stida body. You can see here in the picture, this is stida body, and these are residual body of sporocyst, and this is residual body in oocyst. Each sporulated oocyst contains 4 sporocysts, and each sporocyst contains 2 sporozoids. These are 4 sporocysts. Okay, you can see here 1, 2, 3, and 4. These are sporocysts. Okay, and these 4 are sporocysts. Okay, and it contains sporozoites. Each sporocyst contains two sporozoites, okay? So these are four sporocysts, and each sporocyst contains these two sporozoites, okay? One and two, one and two, one and two, one and two, okay? I hope you got this structure. So this will remain the same, micropyle and micropylar cap. Only difference in sporulated oocyst is the presence of these four sporocysts and the sporozoites. And this is data body, right? Now, Eimeria species found in the poultry. Okay, these are the species, these are the site of in-host and what disease it causes. So first we talk about the Eimeria tenella. So site is cecum and it causes sickle coccidiosis. Tenella causes sickle coccidiosis. Now Eimeria necatrix. Site is in small intestine. It causes intestinal coccidiosis. Okay, remember necatrix causes intestinal coccidiosis and tenella causes sickle coccidiosis. Eimeria bruneti site is rectum and it causes rectal coccidiosis. And Eimeria acervulina, again in small intestine, cause intestinal coccidiosis. Eimeria maxima, again in small intestine and intestinal coccidiosis. Eimeria mitis, intestine and intestinal coccidiosis. And paracox, intestine and intestinal coccidiosis. So you have to remember these two, tenella and bruneti. Tenella cause sickle coccidiosis and bruneti cause rectal coccidiosis. The rest of these cause intestinal coccidiosis. All right. So let's talk about the Eimeria species in cattle and buffalo. So Eimeria zerni, winter coccidiosis, most common and pathogenic, okay? Eimeria bovis, most common. Eimeria beryli, found in water buffalo. And this is Eimeria buccidonensis, biggest species among Eimeria species in cattle and buffalo. Eimeria canadensis. Okay, so you have to remember this. Zerni, water coccidiosis, is most common and most pathogenic.